In the last video, we talked about amino acids that were nonpolar. In this video, we're going to continue our talking of amino acids and talk about the polar ones. In the last video, like I mentioned, we talked about nonpolar ones, and there were 9 nonpolar amino acids. Well, the rest are polar, so there have to be 11. So there are 11 polar amino acids that we're going to talk about. And we're going to do the same thing. We're going to talk about their names, their abbreviations, their structures, and fun facts associated with them. Excuse me. So let's just get right to it. So polar amino acids are a little bit different than nonpolar ones in that they're subdivided into different categories. Within polar, there are polar neutral amino acids. Whoops. Let me make that in a different color. So there are polar neutral, of which there are 6. There are, actually, let me give myself a little bit more space to draw those neutral ones. So there are polar acidic, of which there are 2, and then there are polar basic, of which there are 3, right, for a total of 11. 6 plus 2 plus 3 is 11 total. So let's get right to it and start drawing these ones out. Okay, so for the polar neutral ones, the first one I'm going to draw looks like this. Remember the carboxyl group up at the top, alpha carboxyl, and then alpha. Oops. Oh goodness. Sorry about that. I meant to put an H there. Okay, the alpha amino group, the hydrogen, and then the R group. So this first R group looks like this. I have a CH2, and then a C with an amino group, and a carbonyl group. Okay, this amino acid is called asparagine. Oops, that's an I in there. Okay, and asparagine, its 3-letter abbreviation is ASN. Okay, and its 1-letter code is N. Now, it's not ASP. We'll actually get to there actually is an amino acid that starts with an ASP, or excuse me, that also starts with an ASP, and its 3-letter code is ASP. That's actually called aspartate or aspartic acid, which we'll draw here in just a moment. But this is asparagine. Okay. Asparagine looks a lot like this next amino acid that I'm going to draw, which looks like this. And again, I'm going to do the same thing I did in the last video, where I'm not going to draw these 3 groups here. It's sort of implied that they're going to be there, although the correct drawing would include all of those different substituents. The next one looks like it looks pretty much identical to asparagine. The only difference is that there's one more CH2 group. So there's still this group here. Oops. Okay, this is called glutamine. And glutamine's 3-letter code also ends in an N, just like asparagine, so except it's GLN. And its 1-letter code is Q, right? Because G has already been taken by glycine. So, and if you think about it, a Q kind of looks like a G almost. I don't know if that's helpful, but you can go sort of think about it that way. Okay. Both of these amino acids have this particular functional group here, and that is called an amide. Okay, so these both have amide or amide functional groups. Those groups are not ionizable. Okay, ionizable, not ionizable. They cannot be turned into ion. They cannot be protonated or deprotonated at all. Okay, it's important to keep that in mind for those two. Okay, so let's draw a few more of the polar neutral ones. Okay, so this next one will look like this. It'll just have a CH2 group and an OH. Excuse me again. That's called serine. S-E-R-I-N-E. 3-letter code is S-E-R. 1-letter code is S. And that's it. Okay. Now, the next one, and the reason why I haven't mentioned any particular fun facts about this one yet is because I want to write a collective fun fact about a few that are related to serine, much like I did here with asparagine and glutamine. They both have the amide groups, so I sort of associated them both with that particular little characteristic there. So serine, and I'm going to write out this next one. This is has a CH2 and a phenyl group. So far that would be phenylalanine, but then we're going to tack on this little OH at the end here, and then what we get is tyrosine. Okay, now tyrosine is TYR, and its 1-letter code is Y. Okay. Notice these both have OHs, which is what I'm getting at here in just a moment as far as the fun facts. Notice though that tyrosine has this benzene ring, right? So that is aromatic. Okay. One more of these amino acids. So that's 4 so far. There are 2 more that we want to draw. The next one that also has an OH group looks like this. Has a CH and then an OH and a CH3. This thing, I'm going to scroll down just a bit, make some more room. This is called threonine. Its 3-letter code is THR. So there we go, back to the first 3 letters. Its 1-letter code is T, so that's why tyrosine and tryptophan don't have T, right? Because threonine took it, so they had to get specific ones to them. And we already talked about tryptophan, but tyrosine here as a Y. Tyrosine has a Y in it. That can sort of make sense. Anyway, so why did I write these 3 like this? The reason why is because they all have an OH group, right, a hydroxyl group, in their side chain. And the reason why that's important is because all 3 of these, okay, I'm going to number them, 1, 2, and 3. Okay, I guess I might as well number those 4 and 5 in just a moment. I'm just going to do it now, even though I wrote them first. 1, 2, and 3. Oops. Write it here. 1, 2, and 3 can all be phosphorylated in proteins. Why is that important? Okay, first of all, they can all be phosphorylated in their proteins due to having that hydroxyl group. Okay, that hydroxyl group is the group that actually gets phosphorylated. A phosphate group gets replaces that hydroxyl group. And the reason why that's important is that is important in regulating protein activity. And we'll talk about this of this idea in future videos, regulation of protein activity. Okay, sometimes phosphorylating a protein could activate it, sometimes phosphorylating a protein could deactivate it, sometimes, and vice versa for actually both of those. It sort of depends on the protein itself, but the whole idea is that phosphorylating an amino acid residue of one of these 3 amino acid residues on a protein alters its structure. And something that we learn in biology, biochemistry, just in science in general, is that structure determines function. And if we alter the structure of a protein, chances are we're going to alter its function. So phosphorylating a protein could make it more active, less active, or totally inactive, or completely active, whatever it may be. So it's important to keep that in mind for these 3 different amino acids. So that's 5 of the 6 polar neutral ones. Let me draw this last one down here. Okay, the last one looks like this. Okay, it looks like serine, except instead of an OH group, it has an SH group. Actually, before I continue on with this one, I wanted to mention one last fun fact about threonine, and that's that it also has 2 chiral centers. I forgot to mention that. I'm sorry. It has 2 chiral centers, right? This here and this here, right? Because they both have 4 different attachments. So anyway, sorry about that. Back to this one, this last one here. This one is called cysteine, and this is a very important amino acid. It's one that you should be very familiar with. It's a pretty simple structure, but it's a very important amino acid. Its 3-letter code is CYS, and its 1-letter code is C. In the last video, we mentioned that methionine as being one of the 2 sulfur-containing amino acids. Well, cysteine is the other one, right? So I guess I should write 2 of 2 sulfur-containing amino acids, right? And this SH group is called a thiol. Okay, and what ends up happening is this thiol group, if it's next to another thiol group, it can form disulfide bonds or disulfide bridges. That's very important. Okay, disulfide bonds, also called disulfide bridges. Okay, and the reason why that's so important, we'll talk about shortly. Maybe not in this video, obviously, but in a future video. Disulfide bonds are covalent bonds. Just to give you a quick idea of how they look, they kind of look like this. If you imagine 2 SHs being next to each other, right? There's an SH there and an SH here. What happens if they come together? They can form, if those hydrogens go away, and I'll talk more about exactly how this happens. We can form a covalent bond between the 2 sulfurs, and that actually is the disulfide bond, or the disulfide bridge. So that we'll talk about that and how it affects the protein structure and why it's important and all that sort of stuff later. But I just wanted you to be familiar with these ideas. But keep in mind that cysteine is a very important amino acid to remember and to be familiar with. Okay, so that's all the polar neutral ones. So I guess I should put 6 right here, right? So that's 6 total. Let's talk about the acidic and basic ones now. Scroll right back up. Okay, so let me separate these really quickly so we don't get too confused. Okay, so polar acidic. Now the polar acidic ones, they're going to have side chains, right, that have an acidic functional group, specifically a carboxylic acid functional group, right? So these ones here, oops, I don't want to put that in white. Let me write it in green. So these polar acidic ones, they're actually going to look very similar to asparagine and glutamine. So the only difference is that instead of having amide groups or amide groups, they're going to have carboxylic acid groups. So asparagine had 1 CH2 and then that amide group, right? The difference here is that instead of having an amide, we're going to have a carboxylic acid, right? And a carboxylic acid at a neutral pH, carboxylic acids typically have pHs, or excuse me, pKa's, that are pretty low, and they're acidic, right? So at a neutral pH, they will likely donate their proton and exist in their conjugate-based form like this, right? So this is actually aspartate. This amino acid is called aspartate, also called aspartic acid. So I'm going to write here aspartate, okay, because that's the way I drew it in, right? ASP which is also, and its one letter code is D. Okay. Um, it's also called aspartic acid. Now, the reason why I wrote aspartate first is because it's in its conjugate base form, right? So because it's in its conjugate base form, it carries that ate name. It's the conjugate base that we're talking about here, right? If this portion here was protonated, it would be considered aspartic acid. Although, like, I mean, the technicalities are not really too important, but in case you're wondering why there are two names, that's the reason. So notice that at a pH of 7, right? Again, I'm drawing all of these at a pH of 7. The polar acidic ones will carry a negative charge, okay, at a pH of 7. And this is why, because this negative and this positive balance out to 0, and then all you're left with is this negative charge here, okay? So overall, it carries a -1 charge. Now, the other polar acidic amino acid looks a little bit like glutamine. It's very much the same way as aspartate or aspartic acid worked. It looks exactly like glutamine, except instead of an amide group, it has a carboxylic acid group. So CH2, CH2, and then C double bond O, that minus there, and then we have glutamate. So glutamate, right, or glutamic acid, okay, this one is GLU. It's also E. I don't know, so I remember glue for glutamate. In any case, this one also carries an overall negative charge, that amino acid. Okay, so the way I remember these structures and these structures here is just I remember the similarities between them. Oftentimes when it comes to memorizing all these different amino acids, all the little fun facts about them, their structures, their names, all that sort of stuff, a lot of the times it helps to sort of group things based on similarities. So knowing that how these two polar acidic ones are related sort of to these polar neutral ones in structure, that can kind of help you remember what the structures are like. Okay. Well, that's pretty much it for the polar acidic ones. So let's talk about the polar basic ones. So the polar basic ones. Okay. Now the polar basic ones, they will typically carry a positive charge at a neutral pH. Okay. There is a slight exception, and I'll go over that. But you can imagine that if these polar basic ones have side chains with amino groups when they're protonated carry a positive charge, right? So if we have an alpha carboxyl group and alpha amino group, right, those charges are going to balance out. If we're left with a positive charge, amino group that's charged, we're going to have a positive charge. So let me draw out the three polar basic ones. The way I kind of remember the polar basic ones is I think of I think HAL. So HAL is a pretty basic name, okay? Basic name. So the three basic amino acids are histidine, arginine, and lysine. Okay, so if that helps, just to help you remember the three basic amino acids. Now these aren't the one-letter codes. This is just to help you to remember the names. And anyway, let's draw the first one, being histidine. So histidine will look like this at a pH of 7. Now, I know I mentioned this positive charge here, but I'm going to draw this first one as an exception. So histidine looks like this, right? Its side chain looks like this. It has a CH2, and then it's connected to this little ring structure here. And this ring has a double bond here, and a double bond here, and an N here, and an N here. The N that does not have the double bond carries just an H, and that's all it's going to carry. However, this one over here, this N that's double bound that is involved in that double bond, can be protonated. Now, at a pH of 7, it isn't protonated. Why? The reason why is because this group has a pKa of approximately 6, right? So if its pKa is lower than 7, then that means it's going to act as an acid at that pH and donate its proton to the environment, so it won't be protonated. However, if you lower the pH enough, this group will be protonated, and it will look like this. So I'm going to put lower pH. Okay. If you lower the pH, then you'll get that group to be protonated, and it will look, instead of like that, it'll look like this. Okay, so it'll have the CH2, and this NH will still be the same. The only difference is that this N will now be protonated and carry that positive charge there. Okay, so these are both histidine, right? Just at different pHs. Now, typically, the basic functional groups will carry a positive charge, and that holds true for arginine and lysine. Histidine is just a little bit weird. This group here, this actual functional group here, is called an imidazole group. Okay, so I'll write that in purple. So this is an imidazole group. Imidazole group. And that's actually the group that has the pKa of 6. Okay, so that's histidine. Let's draw arginine and lysine. So arginine looks like this. Arginine has 3 CH2s and then an NH and then another carbon and then, let me scroll down just a tad so I have a little bit of room here. And then this carbon has an NH2 and this carbon has an NH2 as well. But this N has 4 bonds to it, so it's going to be protonated. Okay, so this here is arginine. Okay, so arginine, arginine. Okay, and arginine's 3-letter code is ARG, and its 1-letter code is R. So arginine, like I think like a pirate. Oh, I forgot to write histidine's name in 3-letter code and 1-letter code. So histidine is spelled like this. Sorry, histidine. 3-letter code is his, and then its 1-letter code is H. Sorry about that. I should have written that earlier. Okay, so this is arginine. Notice again here the double bonded nitrogen is the nitrogen that can be protonated. The pKa of this functional group is actually very, very high. I think it's 12 and a half, close to 12 and a half. This group here is called a guanidino group. Okay, so it's a guanidino group, and it's very, very basic. In fact, it's so basic it has a pKa of, or like I said, around 12.5. Okay. So that's histidine and arginine. There's one more, is lysine, is the last polar basic amino acid. And let's draw that and finish this off. So the last polar basic one will look like this. I have that carboxyl group, amino group, hydrogen, and then lysine has 4 CH2s. A friend of mine once told me that lysine was the amino acid involved in Jurassic Park. I don't know, they talked about it in Jurassic Park, the film. And one of the ways is that it helped them remember the structure of lysine was that a lot of the dinosaurs that were talked about had 4 legs, right? So 4-legged dinosaurs, 1, 2, 3, 4 CH2s. So silly, but if it if it helps you remember the structure, then by all means use it, right? So as I mentioned, this is lysine, and lysine, its 3-letter code is LYS, and its 1-letter code is K. Okay, and that's because leucine already took the L. So again, so notice that for arginine and lysine, they both carry overall positive charges, right? So here for lysine, we have the negative here, the positive here, and the positive here. So that's an overall +1 charge. And in arginine, we have the negative plus and then a plus here. So that's an overall positive charge as well. So that's pretty much it for the polar amino acids. So it's, again, keep in mind these differences, these subtleties, and it'll be very, very important in the future to understand what these amino acids will look like given a particular pH. So if you know the pKa's of the side chains, or all the different ionizable groups, right? So the polar acidic and polar basic amino acids have groups that are ionizable, right? The polar acidic ones can either exist in a protonated form that's neutral, or the base form like this, which is negatively charged. And the polar basic ones, if they're protonated, they'll be positive, and if they're deprotonated, they'll be neutral. So it's very, very important to understand how we can use the Henderson-Hasselbalch equation, which we talked about earlier, to help us understand how these different amino acids will look at a particular pH if we know the pKa's of all the ionizable groups, including the alpha amino and carboxyl group and the side chain pKa, if the side chain has a pKa. Alright, so I hope that was helpful, and that's pretty much all the amino acids. See you next time.